cancer is currently considered to be caused by genes. That's the prevalent view, or genetic changes anyway. So you've conducted the rigorous research, as you said, about the metabolic and mitochondrial theory or origin of cancer. So can you tell about how the mitochondria and mitochondrial metabolism contribute to cancer?
Yeah, well, first, you're correct. Right now, the current view of that cancer is a genetic disease under what we call the somatic mutation theory, which is worldwide to be thought of in all major Western societies, based on the NCI, National Cancer Institute, and most of the cancer institutes of all major Western countries will say that cancer is a genetic disease, and this drives the research as well as the treatments. It's important to recognize that the second major theory, which is the correct one, well, that is, cancer is a mitochondrial metabolic disorder based on the mitochondrial metabolic theory of cancer. So we have two major competitive theories that have attempted to explain the origin and progression of cancer. Okay, what I have done is looked back at the work of Otto Warburg, the German scientist, to determine whether his ideas might be more accurate to explain the origin of cancer than the current somatic mutation theory of cancer. And let's look at this. Once you realize that the accuracy, which of the two theories can better explain the origin and progression of the disease, should determine then the ways that we try to prevent or manage the disorder. Okay, so today you have to realize worldwide cancer is out of control. We are not dropping death rates in any area of the world. Cancer is increasing in all major populations. So clearly, the last 50 years thinking that cancer is a genetic disease hasn't led to any real improvements in overall progression-free or overall survival. So, and according to the somatic mutation theory, the accumulation of mutations in the nuclear DNA is responsible for the dysregulated growth of these cells. So let's consider that for a minute. Now, what is often not, before I get into why it's a mitochondrial metabolic disease, let's look at the shortcoming of the dominant theory that says it is a genetic disease. So according to the somatic mutation theory, somatic mutations, which mean mutations in the DNA of cells in a tissue, as opposed to germline, something that you would inherit. Okay, so somatic mutations cause the dysregulation of cells in a particular tissue or organ, and leading to cancer. But what is not discussed is that there are some cancers where mutations have not been found. Yet we have dysregulated cell growth, and people don't even know about this. They either choose not to know about it. Then they broke down, not all of the somatic mutations are actually contributory towards dysregulated cell growth. So they've made a different group of mutations called driver. These are called driver gene mutations. They drive the dysregulated growth because they are thought to play an important role in some aspect of cellular biology. Okay, so now over the last few years, more and more research has been found showing that we find all of these driver mutations in normal tissues of our body that have no dysregulated growth. So this is kind of unusual. How do you explain some tumors that have no mutations and then normal cells that have driver mutations that never become cancer? This is, the field says, we haven't figured that out yet. We're still trying to close the knowledge gap. rather than saying, maybe we're wrong. Maybe cancer is not a genetic disease. Oh no, they won't accept that. So, and then we know from the nuclear mitochondrial transfer experiments that when you take the nucleus of a tumor cell and move it into a new fresh cytoplasm, you have regulated growth. And if you take the nucleus from a normal cell and put it into a tumor cell cytoplasm, you have dysregulated growth, completely ruling out a nuclear-driven process to dysregulated cell growth. All fingers pointing to something in the cytoplasm of the cell, and that something is the mitochondria, the organelle that generates energy and metabolic homeostasis within the cell. Okay, so now we have the mitochondrial metabolic theory, which was originated with the work of Otto Warburg and has been since expanded upon by us, correcting a lot of the misunderstanding that was associated with Warburg's original findings. Warburg said all cancers originate from a disruption of oxidative energy metabolism, number one. And number two, this was coupled by a compensatory substrate level or fermentation, energy without oxygen. So I looked into this for more than 20 years. I did research on this, and I concluded that Otto Warburg was essentially correct. We have never found a tumor cell with a normal number, structure, and function of mitochondria in the cytoplasm, regardless of the genetic mutations. So all major cancers are fermenting. They're getting energy from non-oxidative, non-oxygen mechanisms. This all supports what Otto Warburg said. Now we are cleaning up the mess that was associated with the misunderstanding of Otto Warburg's ideas. And we're showing that oxygen consumption in cancer cell is not linked to ATP, energy through oxidative phosphorylation. Lactic acid production in cancer cells is not an accurate marker for energy through fermentation. And we're starting to say why this is the case and how to improve our understanding. So far, we have been unable to find any tumor cell that can live without glucose and the amino acid glutamine, meaning that all major cancers are dependent on these two fuels for dysregulated cell growth. This is because the energy from oxygen in the mitochondria is dysfunctional. So the cell must go to a non-oxidative mechanism, which is fermentation, driven by two fuels, glucose and glutamine. The mutations in the nucleus of cancer cells are downstream effects of the damage to the energy metabolism. So most of what people say, the world is saying cancer is a genetic disease, and we're saying those mutations are not the cause of dysregulated growth, but the effects of energy metabolism defect. So when you put it in that context, this changes the whole view of how we prevent and manage cancer relative to what we're doing today with all these toxic poisons, immunotherapies, all these kinds of things, which are very, very expensive. They cause harm to the body, and they're not effective for managing the majority of cancers in any country of the world.
Thank you so much of that thorough explanation. Before we go into talking about these therapies and the metabolic therapies, I'll make a brief summary, and please correct me if I'm wrong. So what I'm understanding is that we have confused the chicken and the egg. Instead of genes driving the cancer, there's something that happens in the mitochondria, which is basically that they cannot use oxygen for making ATP that we would usually use in our mitochondria through oxidative phosphorylation. So they utilize this backup mechanism, almost quote unquote, where they produce lactate, where they ferment glucose and glutamine. So why does a cell that ferment glucose and glutamine and use this alternative fermentation mechanism, why does that have a dysregulated growth? Why does it spread like cancer cell does?
Yes, that's an exceptionally important question. Because the organelle that controls the cell cycle and the differentiated quiescent state is the mitochondria. The gradient inside the mitochondria controls calcium signaling in the cell. calcium signaling ultimately regulates the cyclins in the cell cycle. So the organelle that's supposed to maintain the quiescent differentiated state gradually loses grip of maintaining the metabolic homeostasis. And as the cells begin, because if that part of the energy is disrupted, the cell will die unless it can compensate by upregulation of fermentation. If it can't, so there's two things that Warburg said. If you block oxygen energy too quickly, too acutely, you will kill the cell and it can never become a tumor cell. So it's got to be a chronic disruption of oxidative phosphorylation energy through oxygen, allowing the cell time to upregulate these ancient pathways of fermentation. So if a cell cannot upregulate fermentation, it will never become a tumor cell, because all tumor cells are fermenting glucose and glutamine when you examine them. Now, the interesting thing, there are a couple of cells in our bodies that cannot do this, and they are cardiac myocytes, which are the muscle of the heart that's constantly beating, filled with mitochondria. And if you block the mitochondria, the heart muscle stops and you have a heart attack. So it cannot sustain a shift to fermentation and still maintain the same level of metabolic efficiency. The other cell is neurons in the brain. They can ferment, the heart and the neurons can ferment robustly for about 5 minutes, and then they die. They can't gradually compensate for lost oxidative phosphorylation over time. So you rarely, if ever, find cancer involving cardiomyocytes or neurons in the central nervous system. So that tells us, like Warburg said, you've got to have cells that can transition from oxidative phosphorylation to this glucose-glutamine fermentation if you're going to get cancer, which is dysregulated cell growth. And you also have to realize that the default state of mammalian cells is proliferation. And when the mitochondria that control the differentiated state and maintain quiescent gradually lose their ability to produce energy efficiently, the cell enters its default state, which is proliferation, driven by fermentation metabolism, from our knowledge that all cells on our planet were driven by fermentation before oxygen came into the atmosphere 2.5 billion years ago. All the cells were fermenting at that time. In our tissues today, those pathways are heirlooms of the ancient past. And when oxidative phosphorylation gradually becomes dysfunctional, those ancient heirloom pathways now become dominant. And they are leading to this dysregulated cell growth out of control, because the organelle that's supposed to control them is now dysfunctional. They bypass programmed cell death, and they proliferate like crazy, driven by a fermentation metabolism. And just as in the past, there was no regulation of cell growth, this fermentation was associated with unbridled proliferation and would stop only when the fermentable fuels in the environment became depleted, and then the cells would die. So what we have today in cancer, as long as those cells have access to fermentation fuels, which are predominantly glucose and glutamine, they continue to grow, and they're very hard to kill. The simple way to kill them is remove the fermentation fuels and transition the body over to a non-fermentable fuel, like fatty acids and ketone bodies, which help our normal cells but cannot be used by the tumor cells. So we marginalize the energy metabolism of the tumor cell by transitioning away from fermentable fuels to respiratory fuels, which the tumor cells can't use, and they become gradually degraded by our normal health healing processes within the body. It's a beautiful way to manage cancer. It's effective when it's done correctly. The problem is almost nobody knows. What I just told you is not known or cannot be understood by the majority of people that do cancer research or treat patients in the clinics. That's the great mystery. How is it possible that what I just said cannot be either understood or accepted by the majority of people in the field? That's what the strange thing is.
Right. So the current cancer treatments are probably one of the most strenuous, most difficult experiences for a human to go through, can you discuss a little bit about what we have available now, and how is a metabolic management of cancer different from this?
Yeah, well, right now I think everybody knows it's slash, poison, and burn, which is surgical mutilation followed by poisonous chemicals, followed by radiation. Now, here's the interesting thing. Everybody knows those things are medieval. I think during the Spanish Inquisition, the Dominicans that tortured heretics would be quite impressed with what we do to these cancer patients in the form of torturing them. What we do to these poor cancer patients is tragic, based on our lack of knowledge. So basically, what we do is we use these very toxic, aggressive approaches, which harm the rest of the body in many, many different ways. And I know there are many, many cancer survivors who have actually survived mutilations, radiation, and poisonous chemicals. But their bodies are beat up in many different ways. They suffer what we call adverse effects from these toxic treatments, and may suffer later on from recurrence of cancer or neuropsychiatric problems, digestive problems, bone density problems, all kinds of problems, because you survived extremely toxic treatments to manage your cancer. And now, don't forget, we continue to do these because the promise of the genomic projects, the precision medicine, which was supposed to replace these medieval treatments, has never been realized. So we look at immunotherapies today. Immunotherapies are also based on the somatic mutation theory of cancer. So that's the new thing. Opdivo, Keytruda, CAR-T immunotherapy, all these, you hear them advertised somewhere, at least in our country, they're on TV at night. And they spend more time telling you how the treatment will kill you before it actually helps you. So you have to realize, and so you're using drugs that have a high probability of making your life worse. There are people who are doing better, but many of them don't do better. And sometimes these therapies will kill you faster than the cancer itself. What's going on here? Why is this happening? Because those therapies are based on the somatic mutation theory. And if the somatic mutation theory, like I've just mentioned, is no longer credible, then you are not going to see any major drops in death rate, because you're treating patients based on an incorrect theory of what the nature of the disease is. So when you do metabolic therapy, now you're targeting the very essence of what's driving the dysregulated growth, the fuels that are needed for that dysregulated growth. So you take away those fuels or try to interrupt them as best as possible, which can be done by water-only fasting, which can be done by certain drugs and procedures that interfere with the availability of glucose and glutamine. And gradually you bring, and you do it at the same time you are making the rest of the body healthy. You're not harming the rest of the body with metabolic therapy, because we transition the patient away from glucose to ketone bodies and fatty acids, which the normal cells can now use quite effectively, because we evolved to do that. We have to understand who we are as a species. Homo sapiens evolved from other primates about 2.5 million years ago. How did we expand? How did we exist on the planet during a couple of ice ages? What did we do as a species to survive? We were always in a state of nutritional ketosis because there was no highly processed carbohydrates available in the environment at that time. And that was our existence for hundreds of thousands of years. Only within the last 50 or 75 years have we found ourselves confronted by massive doses of highly processed carbohydrates, minimal exercise as compared to the past, and stress and all the things of modern society. So what we do is we recognize who we are as a species, how we evolved, and we try to bring our bodies back into that state of Paleolithic existence, which is reducing blood sugar and more exercise, getting the ketones elevated, and then we can come in with repurposed drugs that we know can target the glutamine in these tumor cells. And we do it very strategically according to our press pulse therapy that we published, which then recognizes that glutamine cannot be targeted aggressively. You have to do it very strategically, because that same fuel not only drives the dysregulated growth of the tumor, but it also makes our immune system, keeps our immune system function. Our gut health requires glutamine. A urea cycle requires glutamine. So you cannot go in and target glutamine too aggressively, because you could end up defeating what your purpose was, to make the body healthy. So glucose is not essential for our existence. We can switch to ketones and fatty acids. The tumor cell absolutely has to have the glucose. And we target the glutamine strategically with pulsing using repurposed drugs. And eventually we slowly degrade the tumor while enhancing the health and vitality of the rest of the body. So, and then you'll gradually see either a stabilization of the tumor. In other words, it just stops growing, never goes away, but it's there, but it doesn't get worse. Or you actually get this thing to gradually go away. And we don't know. Everybody's different. Some people, they live with it. Other people are happy that it went away. We don't know how long it went away for, 5, 10 years. All we know is that we are on the path for a more reasonable way to manage cancer based on our understanding of biology, biochemistry, and evolutionary biology. This is the strategy, not this mindless stuff that we're doing in the clinics today that are not based on the mitochondrial metabolic theory, which is the correct theory to define the nature of cancer. And once you understand that theory, the strategies for managing cancer will be much less toxic and far more therapeutic than what we currently have in the clinics today.
Right. So if there is listeners at this point want to learn about the depth of this theory, I recommend the book Cancer as a Metabolic Disease. It's pretty long, but it's very explanatory. So it slowly reveals all the details or the important cascades about this theory and why it ultimately makes most sense. For me, it was a huge revelation to read that book. And like one thing though that I still keep thinking is that why is the glutamine? So I understand glucose is fermented and is fermentable. Glutamine is an amino acid. Is it just glutamine or is it all other amino acids? Does protein feed cancer? Does that make sense as a question?
Well, okay, let's look at, we interrogated the tumor cells. Here's the way we do it. It's not that complicated. We take our cells and you grow them in culture in rich medium, bovine serum, calf serum, and all this beautiful stuff. You add glucose and glutamine, you have all this cocktail of stuff, and they grow like crazy. They're very healthy cancer cells. Then we take away everything. We take away the serum. We take away the glucose. We take away the glutamine. We take everything away and just grow them in a salt solution, and then time them and see how long it takes them to die. So obviously, with no food, no nutrients, cells can only survive for a short period of time. And then you time them. Mouse cells are all dead in 24 hours. Human cells are all dead anywhere from 48 to 72 hours. Human cells, because our basal metabolic rate in our cells is very slow, much slower than the mouse. So mouse or rat, they die fast. So once the cells then we know, okay, they're dead. Now we add stuff back to these cells. So we grow them in saline, and then we add each individual amino acid back by itself to see what it does. Is there any evidence that a single amino acid can maintain the life and viability? And what we use bioluminescence, which is a marker for ATP, which is energy. So we're looking at energy. We're looking to see what fuel could provide energy for the cell and keep it from dying. And we got all through all 20 amino acids. And yes, glutamine is the number one. There's no question about it. You can get a little bit from glutamate, maybe a little bit from asparagine, but nothing like the power of glutamine. Then we go through and look at all the carbohydrates and we check out. And this glucose, for sure, is the number one carbohydrate that can perk them up. And when you put the two together, glucose and glutamine, you get a powerful synergy between these two fuels that allows the cells to grow quite robustly, even in the absence of serum, as long as you have these two fuels. And then you say, what is the mechanism by which these fuels are producing ATP to allow the cell to grow and live? Because ATP, without ATP, nothing grows. So we know the energy currency is energy. It's adenosine triphosphate. Where does it come from? It comes from glucose and glutamine. How? So Otto Warburg and others clearly showed that glucose produces ATP largely through glycolysis in the cytoplasm. And lactic acid is a waste product for that. What we have shown for the first time with my colleague Christos Xenopoulos from Semmelweis University in Budapest, world expert on mitochondrial substrate level phosphorylation or mitochondrial fermentation, what we found is that glutamine is used to produce energy in the absence of oxygen through the mitochondria with the waste product of succinic acid, which is unbelievable because succinic acid should never leave the tricarboxylic acid cycle, the Krebs cycle. It should stay in the cycle and be used to generate energy through oxidative phosphorylation. But what we found was that glutamine is a fermentable fuel inside the mitochondria. So the mitochondria has two ways to get energy. One is oxidative phosphorylation using oxygen to generate highly efficient energy. And the second is this substrate level phosphorylation, which doesn't use as much oxygen and can be producing energy in the absence of oxygen. And how do we know this? So we took our tumor cells and we grew them in the absence of glucose, in glutamine, under hypoxia, in the absence of oxygen, the absence of glucose, and the cells are still making ATP with glutamine. Where the hell is that coming from? And we're seeing succinic acid produced as a waste product. Warburg knew nothing about this pathway. This is the pathway because you need the glutamine. It gives you nitrogen for the synthesis of DNA and RNA. It gives you energy through the mitochondria in a non-oxidative way. So this now clears up so much misconfusion, misunderstanding about what Otto Warburg was saying. He did not know about this pathway. Most of the people today in the oncology research field do not know about this pathway. We have shown evidence now, unambiguous, that this is another fermentation pathway that no one knew about. And now you have glutamine and glucose as the two drivers of the production of ATP, leading to the continuation of dysregulated growth because the mitochondria's ability to generate energy through OXPHOS has been diminished, falling back on these two fermentation pathways, one in the cytoplasm, one in the mitochondria. And this is very hard to determine because when you look at a cancer cell, you say, oh, look at, he's taking in oxygen and he's making ATP. Of course, it's oxidative phosphorylation. What we have shown is the oxygen consumption in a cancer cell is not used for ATP synthesis. It's used for the production of ROS, reactive oxygen species, which are carcinogenic and mutagenic. So like you said, the chicken and the egg, they cause the mutations in the nucleus coming from the oxygen consumption. Therefore, they are not the cause. They are the effects of the destabilized energy coming from inside the mitochondria. So the field has been chasing effects for decades, and the dead people are piling up, suffering immensely as the result of this misunderstanding of the metabolism of what cancer cells do and how to manage it. So, yes, I just gave you a very deep, I gave you stuff that almost nobody knows about. Yet we've published this stuff and they scratch their head. I don't know what he's talking about. Why don't you look at the experiments and show how we went about and found all this stuff? And then I think Warburg would be very pleased if he were alive today to know what we have done. His original ideas were correct. He just did not have the correct readouts to quantify what we're telling today. Now we understand this. Now we can move forward in managing cancer in a logical and more effective and non-toxic way, allowing people to live far longer. I don't know if it's a cure or not, but all I can say is that they can live longer with a higher quality of life than what is currently happening in all the major clinics throughout the world.
Thank you so much. And this is groundbreaking. And what is lucky about the current society is that anyone can now go and find this research. So I encourage all the listeners who are interested in finding the truth to go and seek out these papers, because they are not something that you will hear from your doctor, probably, I would assume.
Oh, yeah.
And before we go into the treatments and the patient cases and the research on that, I would actually like to go back a step. And we just established that disrupted mitochondria can't use oxygen. They start fermenting glucose and glutamine, and the cell starts proliferating uncontrollably, because mitochondria are important for preventing the proliferation, the excessive one in the cell. So if we go a step back, what causes the damage of mitochondria? What is the root cause of cancer?
Okay, let's first go back a little bit, because we can have normal cells in our body grow very, very rapidly, not using fermentation metabolism. Let me give you an example. The liver, when we have liver is one organ that can regenerate. Okay, it's called liver regeneration. Regenerating liver cells can grow faster than most kinds of tumors. If you take a hepatoma, which is usually a cancer from liver, it's a liver cancer, hepatoma, coming from hepatocytes. They ferment like crazy. They grow in a dysregulated way, and they're fermenting. However, if you cut a lobe of your liver, you can regenerate the liver, and the hepatocytes proliferate very, very rapidly, but it's not dysregulated because they're using oxidative phosphorus. The mitochondria are normal in those cells, so they know exactly how they can grow very fast, and they know when to stop. The hepatoma grows fast and is fermenting, but doesn't know when to stop because the organelle controlling it is defective. So we know that. So this is another thing that's very important to know: the difference between cancer cell division and normal cell division. And what's the difference? One is that the mitochondria are healthy, and the other one is the mitochondria are defective. Now, getting back to your question about what would cause this dysregulated cell growth to happen in some organ and some tissue of a person. Okay, so this was referred to as the oncogenic paradox by Albert Szent-Györgyi, who received the Nobel Prize for his work on vitamin C. He said almost anything can cause cancer, but what is the common pathophysiological mechanism by which all these provocative things might trigger dysregulated cell growth? So he said, carcinogens can cause cancer. These are chemicals, toxic chemicals that lead to cancer. That's why they call them carcinogens. Radiation, people know radiation causes cancer. Intermittent hypoxia or intermittent oxygen deficiency, as Warburg clearly showed, intermittent disruption of oxygen availability. Chronic inflammation can lead to cancer. Certain inherited mutations like P53, Leifrau-Meni, or the BRCA mutation in the breast cancer, any of these can lead to dysregulated growth. And same with older age. Also, oncogenic viruses like papilloma virus, hepatitis C virus. These things all can produce cancer. But what Szent-Györgyi was concerned about, and others, what is the common pathophysiological mechanism by which all these different things, either inherited or from the environment, could lead to dysregulated cell growth, which is cancer? And I have shown in my book and other papers that every one of these provocative cancer factors eliciting all disrupt oxidative phosphorylation chronically, leading to a compensatory fermentation. So whether it's radiation whether it's intermittent hypoxia or oxygen inefficiency, whether it's viruses, carcinogens, radiation, older age, all of them lead to a chronic disruption of oxidative phosphorylation with a compensatory fermentation. Because when we look at the metabolism of all of these tumors arising from anything, they're all fermenting. So that's what the common linkage is. So we have resolved the oncogenic paradox, showing that the common pathophysiological mechanism is chronic damage to oxidative phosphorylation coupled to compensatory fermentation. This is exactly what Warburg suggested, but we showed in all the different forms of cancer that this is exactly what's going on.
Mm. Can the way we eat these days, can the modern diet cause cancer?
Well, I think it's a combination of a lot of things. It's actually very difficult to get cancer, even though we're all afflicted by it. You have to really put yourself in a lot of different hazardous states for your cells to grow in a dysregulated way. You have to realize it's our modern diet and lifestyle that puts us at risk for cancer. We know this because our closest biological relative is the chimpanzee, almost 98.5% similar in protein sequence and genetic profile. They have a BRCA1 gene, just like a female. There's never been a documented case of breast cancer in a female chimpanzee, if you can believe it. So how is it possible that this organism, which is genetically very similar to us, cancer is extremely rare? Also, human tribes living according to traditional ways, cancer is extremely rare. Albert Schweitzer, the physician humanitarian, didn't find cancer in 40,000 people he looked at in these African tribes. Why they're human beings? Why they have no cancer? Because they're not partaking in the great Western diet and lifestyle like we have. So what is it about our current diet and lifestyle in all major Western societies that puts us at risk for cancer? When our Aboriginal ancestors didn't get cancer, Inuits, Eskimos, very rare, all tribes of humans that live according to traditional ways, cancer is extremely rare. So clearly it's not a genetic thing, it's a diet and lifestyle thing. So when you take these folks that were living according to traditional ways and dramatically put them in the Western diet and lifestyle, they get obese, type 2 diabetes, cancer, dementia, high blood pressure, hypertension, all the same stuff that's afflicting us that live in the Western society. So it's clear within 2 generations, you can go from being healthy to being obese and sick. So what is it that's changed? And as I said, our culture, our technology has evolved so much faster than our biology. We are still basically a Paleolithic organism. Our biology has hundreds of thousands of years in a Paleolithic diet and lifestyle. All of a sudden, within 100 years, is thrown into a totally different kind of environment. You also have to realize that many cultures and societies evolved in agronomy, agricultural, natural agriculture, not consumption of highly processed carbohydrate foods that can be transported all over the globe and still appear to be looking good and tasty, whereas this is a very different kind of a situation that existed even 100 years ago. So within the last 50 years, we have become a highly technological. We don't exercise nearly as much as we used to. We're eating large amounts of highly processed carbohydrates. We have a lot more stress, emotional stress, as the result of our advanced technologies, causing people to have all kinds of problems. So lack of exercise, consumption of highly processed carbohydrates, stress, emotional stress that seems to be hammering us, stuck in front of computers, cell phones, and sitting in traffic. And well, you put all these things together and you end up with a greater number of chronic diseases, type 2 diabetes, obesity, cancer, dementia. All of these are the result of our advancing technologies that our bodies have not caught up with yet. We're still living in a Paleolithic. We need to have a lot of exercise. We shouldn't be eating highly processed carbohydrates. We shouldn't be under so much emotional stress. We put ourselves in this new state. And if you understand biology and biochemistry and evolution, you can figure out what you need to do to attempt to mitigate or at least lessen, reduce some of these conditions that will put us at risk. Diabetes has now replaced smoking as one of the top risk factors for cancer. What's going on with this? And if people were really interested in preventing cancer, it would be rare to find an obese person walking around. Obviously, people do not know how obesity puts them at risk for cancer. Many people that are obese have type 2 diabetes. This is the result of systemic inflammation, which damages oxidative phosphorylation in a particular. Also, sleep apnea happens in many obese people, not all obese people. This is intermittent hypoxia, interruption of oxygen efficiency, exactly what Warburg said. So you put all this together, and the cancer mystery is not a mystery. It's exactly what you would have expected, putting us in this new environment. This is not a mystery. This is what we would expect, and this is exactly what we have. A disease out of control because of lack of understanding of biology, biochemistry, and evolutionary biology. Once you understand what I just said, this whole cancer problem is not a mystery, and it becomes very easy to prevent it if people know why they need to do what they need to do. None of this stuff is it's completely explainable.
Yeah, it's almost like look at any disease, any disorder, and any dysfunction in human. It's usually a combination of just very artificial living, whether it's technology, food, or just disrupted circadian rhythm.
Yes.
So, yeah.
Yes. All of that.
Okay, so let's say there is this very unfortunate, I would say, like a brutal destiny that someone is getting cancer, is diagnosed with cancer. So now, can you tell about the work that you have done with the cancer treatment and some of the results that you've gotten, and what's the state of the current metabolic management of cancer?
Yeah, well, we published the Press Pulse therapeutic strategy for cancer. Problem is, unfortunately, the great tragedy is that oncologists and physicians have never been trained in medical schools to know anything about this. So what they do is what they've been trained to do and what they've been educated to do. They follow standards of care for the treatment. And if they never heard of metabolic cancer as a mitochondrial metabolic disease or know about how to manage it, they have no clue. And the poor patients, they go to these physicians, oncologists, and ask them, I want metabolic therapy. And they say, oh, there's no evidence to support this. There's no clinical trials. Where are the clinical trials? Well, how are you going to do a clinical trial when nobody really understands what you're talking about? You have to educate people to run a clinical trial, to know what they're doing, how to do it, and you have to educate the patients themselves. So in metabolic therapy, the patient themselves play a large role. They are empowered. They need to know that their cancer is driven by glucose and glutamine, and they are in control of lowering blood sugar. They can do that. We developed the Glucose-Ketone Index calculator, which is a ratio of blood ketone bodies to glucose in the blood. And when you do water-only fasting, your GKI, glucose-ketone index, goes way down. It goes way down. Okay, that means you're elevating ketones and lowering blood sugar. The tumor cell cannot use ketones for energy, and it's dependent on sugar. So you're lowering the sugar that it absolutely needs, and you're elevating a fuel that it cannot use. All right, but the rest of the body can use it. So this is the first step in the management of cancer, is we need to bring the patient back into a state of metabolic homeostasis. We need to return them to the condition that they would have been in a Paleolithic lifestyle. So we have to lower sugar, elevate ketones, and bring them back into the zone. And once they're in the zone, which could be achieved by zero carbohydrate dieting, water-only fasting, a variety of different things, and it doesn't make any difference whether it's a Mediterranean, pescatarian, vegetarian, as long as you can get your GKI down. And then your body starts to get into this new metabolic state. And now the tumor cells become more and more vulnerable. So then we can come in with low doses of glutamine targeting drugs that really push so many of these tumor cells over the threshold of life to death without harming the body, because the body is now protected and by the ketones, by the state of nutritional ketosis. So we can lower, even if we're using chemotherapy, we can lower the doses significantly, so minimizing toxicity while enhancing therapeutic benefit. And then we can use non-invasive procedures to assess whether or not we're successful. You can use CAT scan, MRIs, PET scan, all these different ways to look at the tumor and see whether or not it's going away, is it getting smaller or whatever. And then you just stay the course and you gradually degrade the tumor while you're enhancing the health and vitality of the rest of the body. We can use radiation, we can use surgical debulking, we can use all of these things. We just have to know when and how to use them at the appropriate time in the overall management of the disease. So we don't go into this like a bull in a china shop, chopping this poor person apart, poisoning them, and doing all this terrible stuff. We can do some of that, but in a more protracted way under the right conditions when the patient now becomes really, really metabolically fit. And then we can go in and we can hammer away at these tumor cells without inducing systemic toxicity. So as I say to everybody, we already have all of the tools already available today at this time to manage cancer effectively. The problem is we do not have trained personnel. We have governments that say cancer is a genetic disease. We have a pharmaceutical industry pushing all this crazy stuff based on cancer as a genetic disease. So when you put all that together, hospitals are making a fortune. The granting agencies from the federal governments are giving money for these genomic mutation screening stuff. Take that money, flush it down the toilet. All of our private societies, breast cancer societies, look at their scientific advisory boards. They're all thinking cancer is a genetic disease. As long as the whole society and all the institutions think cancer is a genetic disease, what I just said is not going to be done because they don't know anything about it. They think cancer is a genetic. So the theory of what the disease is will drive both prevention and management once it becomes understood. It's just that I see it, but nobody else, not nobody, but more and more people are starting to understand what I'm saying. And when you test the hypothesis and look at the evidence, you can't help but say, this is the right way to do it.
Yeah, and to drive this point home, could you maybe share a patient case
when
using this?
Yeah, we've published several. We publish them as case reports in the scientific literature so that others can say, and we get rid of, there's only one person. Oh, that's only one person. Oh, that's only an anecdote. Well, how many damn anecdotes in one person's people do you want to see before you realize something's going on here? And Maggie and Brad Jones, they're doing the documentary on our stuff on metabolic therapy, cataloging all of these so-called flukes and anecdotes, all stage 4 terminal cancer patients that are still alive. So we have Pablo Kelly from England, Devon, England, who's now a 10-year survivor with glioblastoma. Pablo was diagnosed in 2014 with an inoperable glioblastoma. They wanted radiation and chemo. He said, no, I don't want any of that stuff. He has a very interesting story. He's on the web. He tells you about his story. He asked me what I thought. I didn't, at that time, we really didn't have as much knowledge as we have now. But I said, do water-only fasting and see what happens and go into keto and avocados. He has a big stuff, all this stuff. And, they said he had an inoperable tumor. And he didn't do anything except the metabolic thing. And then after 2.5, 3 years, the tumor grew slowly. It didn't aggressively kill him. It grew slowly, and you could see the MRI images. We published it in Frontiers in Nutrition. You can go and read the paper yourself.
Yeah, I'll definitely link it.
Yeah. And you can see the tumor growing. So they did a debulking surgery after about 2 years rather than right up front, because normally they do it right away. Got to get him in there, chop out that tumor. He said no. But the tumor didn't go away. And then he had it debulked. And he kept very accurate daily evidence from his blood sugar and ketones. So we could calculate his glucose-ketone index, which was always quite low, very nice. And the tumor grew very slowly. So he had it debulked, cut out. And then he thought he was out of the woods because now this inoperable tumor became operable. And the surgeon said, we think we got most of it, if not all of it. So he slid a little back to his diet and lifestyle. And all of a sudden, you could see on the MRI that the tumor was growing again. So this put the fear of God back into Pablo, where he then went back in a deep dive on water-only fasting and this very restricted carbohydrate diet. And the tumor stops growing again. So it's very clear. You could clearly see what was going on. So then after another 3 years, he had a second debulking. And then he had a third debulking, which was, I think, earlier this year. And he's still here. He's still with us. He got married. He has 2 kids. Now, interesting, we always laugh about it because they said Pablo has an inoperable glioblastoma that has now been operated on 3 times since he started his metabolic therapy. So is he cured? No. Is he still alive? Yes. Living far, far longer than he would have been predicted had he done standard of care. Then we had another woman, a woman from Cleveland, Ohio, who had metastatic breast cancer spread to her brain. She went to the Istanbul group, Dr. Slocum and Ikisiki, and they gave her 1 month. She failed all the radiation, chemo, and all the stuff they gave her. And then she was rescued. She was in an emergency room there for about 3 weeks or something. But she's in complete remission, and she sends us pictures from her vacations in Hawaii. This kind of thing. There's a Greek patient with lung cancer to the brain who's out 9 years now. Anecdotes. Excuse me. But we can't yet do standard of clinical trial because we don't have enough trained people, and we're still learning how each person is a unique metabolic entity. So we cannot always use the same exact strategy on every person exactly as we would do it for another person. So we modify the treatment as we go through managing the disease. What is the goal here? The goal is how long can we keep a person alive with a diagnosed stage 4 cancer longer than would have been predicted by all of the statistics that we see from cancer survivor data. Okay, so when you get guys with a glioblastoma living 10 years, this is very rare. This doesn't happen very often.
Absolutely.
Same with lung cancer or breast cancer that metastasizes to the brain. When you have cancer metastasis from one organ to the brain, usually that's pretty devastating. But we're starting to get several of these kinds of folks that have all these metastatic cancer. And we do know from our research that I've done here at Boston College, the metastatic cancer cell is part macrophage, heavily dependent on glutamine. So we know how to manage metastatic cancer because we understand the biology of what the nature of the tumor cell is. They need the glucose and glutamine, really, really good. They need that. So we know how to stop or manage this. Do we cure anybody? I don't know. We haven't been doing this long enough. I published a paper on a dog that was mast cell tumor on the face that was cured, but because it died from old age from heart disease. That's when you know you're cured. Like you're a young lady, God forbid you were diagnosed with breast cancer, you did metabolic therapy, and you died at 96 years old from heart disease. You obviously, with cancer never recurring. So therefore you would have said, well, yeah, I was cured. But you don't know that until you die from old age from something other than what you were diagnosed with. So the goal here is not to say we can cure cancer. The goal is to say, can we live far longer with a higher quality of life than what would have been previously predicted based on the current status of how we treat these people? That's the goal. Not to say, oh, I can cure cancer. I don't know if we can cure cancer. All I know is that we can allow people to live a hell of a lot longer. And I think that's a great success when you think about it.
Absolutely. I think any hope in cancer treatment and for cancer patients is highly welcomed. one thing I'm curious about is that we know that lifestyle contribute cancer after this discussion. Can we use our lifestyle, let's say, can we utilize metabolic strategies, intermittent fasting or ketogenic diets, maybe every now and then to prevent cancer? Would that be efficacious?
Oh, absolutely. The problem is the temptation for the foods and conveniences of our current cultures and lifestyles make it very hard to do that. Do we want to go back and live like a caveman? Killing deer and eating whatever you can find, or do we want to recognize that maybe some level of moderation or maybe we can improve our exercise to be more regular. Maybe we can find ways to reduce stress. Maybe we can try to eat locavore. That means foods that are grown locally. It's a new term in the Webster's dictionary, locavore. It means eating foods that are grown naturally in your local environment. No long-term shelf life, this kind of stuff. All natural, organic kinds of things. And I know that's hard to do that. But I just think that, yeah, you could reduce the risk of getting cancer significantly if you know what causes it. Damage to oxidative phosphorylation with compensatory substrate level phosphorylation is the origin of cancer. How would you prevent that? Well, you prevent that by keeping your mitochondria healthy, because it's very hard to get cancer when your mitochondria remain healthy. How do you keep your mitochondria healthy? Regular exercise. Don't eat as much highly processed carbohydrates. Keep your stress management under control. Do whatever you can to feel better. All of these things. Smoking, of course, was always a risk factor in cancer. We try to reduce the risk factors and improve those things that we can improve, and that would significantly reduce cancer. The problem is when people are told that cancer is a genetic disease, they feel that there's nothing they can do. They're fated to be this. This is nuts. This is not true. This is not true. That's why cancer is not a genetic disease. It's a mitochondrial metabolic disease that we largely control.
And that with proper lifestyle, we could prevent.
Oh, absolutely. You can prevent 80% of cancers by just knowing what I said. You can live longer without debilitating these. Because don't forget, you got type 2 diabetes, hypertension, all of these things are the result of bad diet lifestyle. And cancer is one of them. Dementia is another one. Alzheimer's disease, all these kinds of things. They damage the neurons in the brain. The next thing you know, you have early onset Alzheimer's disease and this kind of stuff. So a lot of this stuff is preventable. And for cancer, it's certainly manageable as well.
Yeah. So we kind of answered to this question, what is the best way to prevent cancer? But there was one question from the listeners, which I would like to ask you, which is, are there supplements that can maybe reduce the risk of cancer?
The best way is nutritional ketosis. I know because I know what how that works. It's unbelievable. It's such a beautiful when the cells take in ketones, the energy efficiency of the mitochondria becomes much greater, and reactive oxygen species are reduced. Whether a supplement, I mean, some people drink ketones, but it's basically water only fasting and low carbohydrate, getting a low GKI, however you want to do that, is therapeutic to prevent cancer. As far as popping a pill or eating something, I don't know. Whatever can elevate ketones and reduce glucose is your strategy that can help. With I can't speak to any supplement, maybe a ketone supplement or something like this, but even that, it comes more natural from the natural body itself that will prevent the cancer.
What do you think about screening and things like full body MRI as a preventive method?
I don't know why anybody would want to do that. Why would you subject yourself to any of these kinds of things unless you had a lump or a bleed or something that would be a little unusual? I mean, if you can catch it um, naturally, biopsies, as I've said and I've published, can spread. In some people, can spread the cancer all around. Why we won't want to stick something in screening procedures or this kind of thing? Um, there's we haven't developed them yet, but they're called liquid biopsies, which can screen people for cancer without sticking you or causing any kind of a surgical disruption. These might be the future. There's a lot of false positive and false negative still associated with that. So we just have to wait with that. But as I said, if you try to change your diet and lifestyle, you probably have a better chance of preventing cancer in the first place. So, but again, and we have people in societies that want to live like a caveman. They eat only raw or cooked deer meat or elk meat. They don't eat any carbohydrates. They're pretty rough kinds of people. But that's not the majority of folks that exist. But there's always those kinds that do this. But yeah, so I don't know about supplements. I don't. Maybe there's some things that you could take that don't harm, that can enhance your mitochondria. But whatever it is, that's what you always have to think about. How is that going to enhance my mitochondrial function without causing reactive oxygen species?
Thank you. So, what would be the good places to find more about this theory? Absolutely, your book.
Most of what I have said today is published in open access papers. So anybody says, and I can't, everybody's, the comments that you'll see, oh, he didn't tell you about lymphoma. Oh, he didn't say this. All of everything that I'm saying is in these papers. I can't cover every base right now. I'd be here for 3 days talking to you. But if you read the papers that I published, it covers all of the different cancers. We haven't found any that can that can grow without fermentation. Maybe there's a rare cancer that we never know about, but all of the major cancers are fermentative, dependent on glucose and glutamine. All of them, open access. People can look at the data for themselves. Work with somebody if you don't know. I understand some of it can be quite heavy and biochemical, but there's some parts of it that everybody can understand. Any literate person can read it and say, I understand that. So that's all there. And our work is largely supported by private foundations and philanthropy. So we like it when people donate to Travis Christofferson's foundation or through my Boston College University website. And we keep the research going so we can then provide the future of cancer management the way we think it should be. And we test everything preclinically before we recommend it in human clinical settings. So I vet, I troubleshoot all this kind of stuff. And then we can talk to clinicians and then they can apply it to their patients without fear of harming the patient, knowing that we have evidence that this could be very powerful if done correctly.
Thank you so much, Professor Seyfried, of this conversation today and your contributions to the field.
Thank you very much, Inka. It was very nice to be here.